Immunoglobulin A is an antibody that plays a critical role in mucosal immunity. More IgA is produced in mucosal linings than all other types of antibody combined. Between 3 and 5 grams are secreted into the intestinal lumen each day. This accumulates up to 15% of the total immunoglobulin produced in the entire body. IgA has two subclasses and can exist in a dimeric form called secretory IgA. In its secretory form, IgA is the main immunoglobulin found in mucous secretions, including tears, saliva, sweat, colostrum and secretions from the genitourinary tract, gastrointestinal tract, prostate and respiratory epithelium. It is also found in small amounts in blood. The secretory component of sIgA protects the immunoglobulin from being degraded by proteolytic enzymes, thus sIgA can survive in the harsh gastrointestinal tract environment and provide protection against microbes that multiply in body secretions. sIgA can also inhibit inflammatory effects of other immunoglobulins. IgA is a poor activator of the complement system, and opsonises only weakly. Its heavy chains are of the type I plus or minus. Forms: IgA1 versus IgA2. IgA exists in two isotypes, IgA1 and IgA2. While IgA1 predominates in serum, IgA2 percentages are higher in secretions than in serum. The ratio of IgA1 and IgA2 secreting cells varies in the different lymphoid tissues of the human body. IgA1 is the predominant IgA subclass found in serum. Most lymphoid tissues have a predominance of IgA-producing cells. In IgA2, the heavy and light chains are not linked with disulfide, but with non-covalent bonds. In secretory lymphoid tissues, the share of IgA2 production is larger than in the non-secretory lymphoid organs. Both IgA1 and IgA2 have been found in external secretions like colostrum, maternal milk, tears and saliva, where IgA2 is more prominent than in the blood. Polysaccharide antigens tend to induce more IgA2 than protein antigens. Serum versus secretory IgA. It is also possible to distinguish forms of IgA based upon their location, serum IgA versus secretory IgA. In secretory IgA, the form found in secretions, polymers of 2 to 4 IgA monomers are linked by 2 additional chains. As such, the molecular weight of sIgA is 385,000 D. One of these is the J chain, which is a polypeptide of molecular mass 15 kD, rich with cysteine and structurally completely different from other immunoglobulin chains. This chain is formed in the IgA-secreting cells. The oligomeric forms of IgA in the external secretions also contain a polypeptide of a much larger molecular mass called the secretory component that is produced by epithelial cells. This molecule originates from the poly-Ig receptor that is responsible for the uptake and transcellular transport of oligomeric IgA across the epithelial cells and into secretions such as tears, saliva, sweat and gut fluid. IgA activity. The high prevalence of IgA in mucosal areas is a result of a cooperation between plasma cells that produce polymeric IgA, and mucosal epithelial cells that express an immunoglobulin receptor called the polymeric Ig receptor. pIgA is released from the nearby activated plasma cells and binds to pIgR. This results in transportation of IgA across mucosal epithelial cells and its cleavage from pIgR for release into external secretions. In the blood, IgA interacts with an Fc receptor called Fci plus or minus Ri, which is expressed on immune effector cells, to initiate inflammatory reactions. Ligation of Fci plus or minus Ri by IgA-containing immune complexes causes antibody-dependent cell-mediated cytotoxicity, degranulation of eosinophils and basophils, phagocytosis by monocytes, macrophages, and neutrophils, and triggering of respiratory burst activity by polymorphonuclear leukocytes. sIgA primarily acts by blockading epithelial receptors, by sterically hindering attachment to epithelial cells, and by immune exclusion. Since sIgA is a poor opsonin and activator of complement, Simply binding a pathogen isn't necessarily enough to contain specific epitopes may have to be bound to sterically hinder access to the epithelium. Immune exclusion is a process of agglutinating polyvalent antigens or pathogens by cross-linking them with antibody, trapping them in the mucous layer, and/or clearing them peristaltically. The oligosaccharide chains of sIgA or IgA1 registered trademark S secretory component can associate with the mucous layer that sits atop epithelial cells. Production of sIgA against specific antigens depends on sampling of M cells and underlying dendritic cells, T-cell activation, and B-cell class switching in GALT, mesenteric lymph nodes, and isolated lymphoid follicles in the small intestine. Transport. Polymeric IgA is produced by plasma cells in the lamina propria adjacent to mucosal surfaces. It binds to the polymeric immunoglobulin receptor on the basolateral surface of epithelial cells, and is taken up into the cell via endocytosis. The receptor IgA complex passes through the cellular compartments before being secreted on the luminal surface of the epithelial cells, still attached to the receptor. Proteolysis of the receptor occurs, and the dimeric IgA molecule, along with a portion of the receptor known as the secretory component, are free to diffuse throughout the lumen. In the gut, it can bind to the mucous layer on top of the epithelial cells to form a barrier capable of neutralizing threats before they reach the cell. Pathology: decreased or absent IgA, termed selective IgA deficiency, can be a clinically significant immunodeficiency. Neisseria gonorrhoeae, Streptococcus pneumoniae, and Haemophilus influenzae type B all release a protease which destroys IgA. IgA nephropathy is caused by IgA deposits in the kidneys. It is not yet known why IgA deposits occur in this chronic disease. Some theories suggest an abnormality of the immune system results in these deposits. Celiac disease involves IgA pathology due to the presence of IgA anti-endomysial antibodies. Inoka Eurosha paragraph NLEIN purpura is a systemic disorder caused by deposits of IgA and complement component 3 in the small vessels. HSP occurs usually in small children and involves the skin and connective tissues, scrotum, joints, gastrointestinal tract and kidneys. It usually follows an upper respiratory infection and resolves within a couple weeks as the liver clears out the IgA aggregates. See also, List of target antigens and pemphigus, TGF-beta, references. External links, Immunoglobulin A at the U.S. National Library of Medicine. Medical subject headings.